We're going to go live. Make sure that this works. Okay, so we're hoping that technology is on our side tonight. So we're really excited to have Dr. Alyca Halladay from the Autism Science Foundation kicking off our webinar series for 2019. Alyca is the Chief Science Officer at the Autism Science Foundation, where she oversees the scientific activities, grants, and initiatives of ASF. She received her PhD in biopsychology from Rutgers in 2001 and completed a postdoctoral fellowship in pharmacology and toxicology from the University of Medicine and Dentistry of New Jersey in 2004. She still holds a faculty appointment at Rutgers. So, and tonight we have Dr. Halladay on talking about Autism BrainNet and why it matters. So welcome, Dr. Halladay.
Thank you, Vanessa, and thank you for everyone from the Dup15q community for joining in at 7 or 8 or 5 p.m. depending on where you live. I'm so pleased to be able to give this webinar. I know how in demand Dr. Dup15q is. I've been trying to get an appointment for my daughter there forever, so I wasn't about to encroach on his presentation time at the meeting in Houston. But I enjoyed meeting all of you at the different events, and I'm really thankful to have this opportunity to kind of touch base and give you an update about what's been going on with brain tissue around the science and just the different aspects of the program. So I wanted to kind of point out brain tissue donation can be kind of a taboo subject, and I completely understand why. But there's a lot of people, a lot of diverse people in this world and in this country that have something in common. And these range anywhere from race car drivers to professional wrestlers to mathematicians to writers. These are all people on the left. You have Dale Earnhardt Jr., Temple Grandin, and Mary Lou Henner, who have said that they would like their brains to be donated to science and research after they die. And then you have people like Walt Whitman and Muhammad Ali and China, who's a professional wrestler, who have actually donated their brains to research for different purposes. In the case of China, who's the wrestler, and Aaron Hernandez, who has quite a checkered history, they have donated their brains to concussion research. They basically understand that the only way that scientists are going to understand what goes on in the brains of people with different psychiatric issues and neurological issues is to study those brains. But why is it really important to study it? Now, with autism and Dup15q and autism, the brain is really the primarily affected organ. It's least studied directly. I know that there's efforts to look at anything between, the spinal cord, the gastrointestinal system, some of the endocrine systems in these disorders, but really we need to study the brain. We can improve, or scientists can improve, diagnosis of autism and predict the course of symptoms. Science is seeing this with disorders like Alzheimer's disease, where because there are so many people that have donated their brains after suffering from Alzheimer's disease, scientists can really, and clinicians can really track the prognosis from the very early, very early warning signs all the way to the late stage dementia parts. Also, there's been enormous understanding of medical comorbidities, especially things like epilepsy and seizures in people with autism and people without autism by studying the brain. It can also understand and help elucidate the causes of autism, what's going on in the brain, and also in rare genetic disorders, and I'll get into some of the overlaps between the rare genetic disorders and autism. And then most importantly, I think for people who are on the phone and on the webinar and interested in this, is it's been helping identify treatment and intervention targets and strategies. And I'm going to get into what those scientific findings have been during this webinar. And I hope I can explain them to you in a way that's easily digestible, because I know we only have an hour, but I'm happy to answer any questions. And actually, we would love for this community to kind of join in and get our quarterly newsletter, and that way you can kind of keep up to date in smaller tidbits than this webinar. The brain is pretty amazing. there are 100 billion neurons and 100 trillion connections in the brain. This is just a kind of a sampling. I mean, I think I can see 5 brain cells in here when there's really 100 billion in the brain. And they start, the connections, these all brain cells all connect to one another. The cell signal starts in what's called the cell body. It travels down something called the axon, and it ends in the synapse. And the synapses are what connect the different brain cells to each other. And what's amazing is that the brain cells can connect in short distances, like tiny bits of a millimeter, all the way to one side of your brain to the other. So there are long-term connections and short-term connections and everything in between. So you can imagine the enormous complexity involved and, essentially how things can go right as well as how things can go wrong, especially in rare genetic disorders. So this is a finding, one of the actual more consistent findings associated with autism, and this is autism, which is called idiopathic autism, where there's no known genetic kind of definition. People in Dup15q community, you guys have a genetic kind of signal that is involved in your family member's autism. There are a lot of people in the community that don't have that. So some of the earliest but also most replicated brain findings come from the lab of Manny Casanova, and he's with me in this picture on the top right-hand corner, that if you look at how brain cells, and these little dots are all the cell bodies that we just talked about, that you may not be able to see this. I didn't see it when I first looked at it. But actually, if you look at how these cells are connected to each other in the cortex of the brain, they actually are structured in columns. And these are called cortical mini columns. And so, they're a certain width apart. And this means that there's a certain amount of distance in between them for signals to get processed. In the non-diagnosed person, and this is, sorry, the bottom one is someone who doesn't have autism, you can see they're also structured in columns, just like they are in autism. But in autism, the columns are kind of spaced farther apart. And again, this isn't something that I think if I looked at this and was able to say, okay, well, what's going on here? This takes enormous sophisticated computer technologies. But it really is one of this idea of a cortical mini column is actually one of the more established findings across brain development. And also, it's also something that has been shown to be altered in schizophrenia. So in autism, you can see that they're just a little bit wider apart. Another consistent finding within autism is the idea that there are too many connections in the brain. And so on the left here, and this comes from the lab of Dr. Jeff Hutzler, who's now in Reno, Nevada. This is, again, you can kind of recognize this from the first part of the presentation where I showed you the cells themselves. Here's the cell body, here's the axon, and here's the synapse. At this part of the synapse where the cells connect to each other, in someone who doesn't have autism, you can see these little tiny sprouts. And actually in the fall in New Jersey, I drive around, I'm kind of a freak, and I drive around and I see these trees without leaves on them, but I see these, or in the spring, and then I see these little tiny sprouts on them, and it reminds me of this. In autism, there are more of these little sprouts. Sometimes more isn't always better. It's thought that perhaps in autism, too many of these connections cause the brain to just be overwhelmed from all the signals it receives. So these are some of the findings we know about autism in general. Luckily, we know we've been able to go, or scientists have been able to go even deeper in terms of studying some of the genetic syndromes associated with autism, and that includes Dup15q. So for example, Dup15q, I'm not going to tell you, you know all this already. It's a genetic disorder, and it's caused by different mutations in the particular region in chromosome 15 on the Q end. Now that's the DNA. So you know the DNA is a double helix. The RNA is when you split that double helix apart, then chemical tags kind of make sure that it can turn the DNA into a protein, which is the whole purpose of having DNA, is to create different chemical signals for different biological events. So really the DNA is the hardwire, the RNA is the activity. So people have already kind of understood what goes on in the DNA, right? It's mutations of chromosome 15, but then how does that translate to expression of proteins? One of the most interesting findings comes from the lab of Dr. Dan Geschwind, and I'm not going to get into too much about this, but if you look at the correlations here, and the reason I'm not going to get into it, and I'll talk about this in a minute, is because he's already done an amazing webinar on this and given really these findings the attention and detail that they deserve. But if you look at the correlations between Dup15q on the left and ASD on the right in terms of some of this genetic activity in the FC, which is the frontal cortex, the TC, which is the temporal cortex, and the CBL, which is the cerebellum, they're actually on the same level, right? So the numbers are very, very tight here. The p-values are very high. That means that the way that the RNA is expressed in people with Dup15q and autism and without Dup15q and autism is pretty similar. So while we talk about, and we will talk about how Dup15q with autism is very different phenotypically from autism without a known cause, actually in the brain, the gene transcription outside that chromosome 15 region is very similar. So of course, within the chromosome 15 region where you see the major mutations, they're very different. But then outside that region, you kind of wonder, well, what is the effect on genes outside that region? And in fact, scientists are finding that it's actually very similar. So Dup15q autism can be very similar in the brain to those without Dup15q and autism. And this is another example of this. The blue line just shows that the changes, while they're similar, do seem to be just a little bit more striking and a little bit more, for lack of a better word, severe. So they're very similar in the patterns, but the slope, which means the intensity of the changes, seems to be a little bit more severe or more intense. So it is similar, and this is Dan Geschwind on the right. It is similar, but it does seem to be a little bit more intense. And this is a flyer at the webinar that Dr. Geschwind gave, which was a collaboration between the Dup15q Alliance and the Autism BrainNet, is on the Dup15q website. If you want to watch it, you can speed through the parts that aren't as interesting and go to the parts that are interesting, although it's all interesting. But I'm not really going to dwell too much on those findings just because he does them much better justice than I could. Now, just because this is about genes, or that some of the studies have been done in genes, and Dup15q is a genetic disorder, does not mean that there's not room for the environment. And the environment, as most scientists think of it, can be very broad. It can be early pharmacological or toxicological. It can even be environmental stimulus. It can be things like socioeconomic status. It can be diet. So it's very broad. In the case of PCB, in the case of, Dup15, researcher, and this was like in 2015, Janine LaSalle at UC Davis, who does a lot of research in Dup15q, you probably have all heard her name, looked at the brains of people with autism, and she kind of looked, she was actually doing a completely different study, and she looked across different PCBs, and PCBs stand for polychlorphenyl chlorinated bisphenol. Sorry, I am a toxicologist, I just blanked on that. This is a chemical that was used in the manufacturing process in the 60s and 70s, and they found that it caused cancer. So the EPA 20, 30 years ago did ban PCBs, but because it's this chemical that doesn't degrade, it's still found, believe it or not, in drinking water, in ground soil, and people are still exposed to it in the environment. It's pretty pervasive and it's pretty nasty stuff. For whatever reason, and she really didn't understand it at the time, it seemed that the brains of people with Dup15q and autism had a much higher level of these PCBs in their brains than people with autism without this Dup15q mutation. And she really couldn't figure out what was going on. Most of the people who had these high levels of PCB were born, before or during the 1970s. So yes, it is the main levels of it is going down over time. But why would there be just more PCBs in the brains of people with Dup15? This started an entire kind of research portfolio specifically looking at how mutations in that area of chromosome 15 interact with environmental exposures. And her next set of studies actually created cell lines with similar mutations in the Dup15q region in cells as people with Dup15q, because she wanted to do things like expose cells to different environmental factors, which you really can't do in people, but you can do them in brain cells manufactured in a dish. And what she found is when she put the cells by themselves versus the cells which had the mutation with PCB in them, they created, they had a completely different epigenetic profile, which I'll talk about in a minute. And they also had, and I'm sorry, I'm going to skip back and forth, they also had a spontaneous mutation in a completely other gene, which was chromosome 22. And so this spontaneous mutation was completely surprising to her. And she said, all right, well, what is going on? So here you have the cells with the Dup15q mutation, the cells with PCB, and then the cells with treated for different periods of time, and then the ones with this particular mutation associated with it, this Dup15q associated with it. And she found, and I think that in fact, here's the spontaneous mutation, which shows a gene-environment interaction. And this doesn't prove, but it does suggest that people with the de novo Dup15q mutation, if they're exposed to some sort of environmental exposure, that may result in other rare mutations as well. She also found that their gene expression or their epigene expression was a lot different. So you take cells that hadn't been exposed plus cells that had been exposed, and the gene expression associated with some genes associated with signaling, secretion, ionic channel, which is how cells communicate with each other, how cells turn on and off, all of these different factors were actually changed. So what this kind of says is that just because it's a genetic mutation doesn't mean that it's impervious to environmental factors. Now, PCBs, along the list of things I think we should be worried about in the environment, PCBs are probably down on the list, not to the bottom of the list because they're still around, but I certainly think that there are more pervasive environmental factors and not just toxicological or chemical exposures we should be worried about either. The problem with some of these studies, of course, is that you kind of have to study them one by one, but by understanding how these things work in Dup15, you don't have to go, we can look at the mechanisms and maybe be able to shorten the list and think about what the mechanisms are a little bit more carefully. So I'm going to skip some of the boring details about epigenetics, although I will say that I just want to point out that your genes are just one part of what makes you who you are. You also have these things on your DNA called methyl groups, and they turn on and turn off your RNA expression. So these things are almost just as important as genetics, but they really don't get the due that they're really worth. So another thing I talked about at the beginning, how Dup15 is kind of one of many genetic syndromes that are associated with autism, or you knew that already. So this is a study looking at a protein called Fragile X protein. Fragile X is another rare genetic disorder associated with autism, where kids and adults, who have the mutation do not express any Fragile X protein. They experience severe intellectual disabilities. and they have features similar, they have a high prevalence of autism. So someone said, all right, the theory is that it's potential. One of the theories could be that rather than working independently, some of these rare genetic disorders across the autism spectrum, and that includes Dup15q, that includes Phelan-McDermid syndrome, that includes Fragile X, and some of these other rare genetic diseases that are popping up with a high prevalence of autism, maybe they're not working separately in silos. Maybe there is a common molecular, genetic, or neurobiological substrate tying them all together. So researchers at the Institute of Basic Research in Staten Island looked at Fragile X protein. They said, is it reduced in people without autism? With autism, but no Dup15 mutation? And then what about people with Dup15 and autism? And as you can see, it's reduced in both groups. And so really, this tells me, so this is, you can't really see it unless you're this is how dark it is in people without autism. This is con for control. And then it's significantly lighter in those with Dup15q and autism. Here's another, if you focus in the microscope, you can see these red dots is the Fragile X protein. And then in people with Dup15q, you can see they're lighter and they're fewer. So it's very possible, and again, this really needs further study, but it's very possible that certain proteins pervade multiple genetic disorders and go across multiple genetic disorders, which could be, in the grand scheme of things, good news, because it shows that if what is working for one rare genetic group could work for other rare genetic groups. So there's been a lot of excitement from the drug industry on treating Fragile X with a number of different therapeutic agents. If it works in Fragile X, it might show hope in people with Dup15 because there seems to be this common neurobiological mechanism where the Fragile X protein is lower. So if you can raise the Fragile X protein in Fragile X, can you also raise it in Dup15? And does that ameliorate some of the signs of autism? So that was, that's been exciting, and I think it's really helpful, but again, it's not the full story. And Vanessa can tell you a lot more about some of the, the clinical drug trials within Dup15 and the targets, but it does offer hope for communities across these rare genetic disorders associated with autism. Now, one of the other things that people are looking at is how changes occur across the lifespan. And the same group, the group at Institute of Basic Research, and here's a picture of them in their labs. This group has really been focusing on what goes on in the brains of people with Dup15q along with the Geschwind Lab. And they're finding an accumulation of what's called beta-amyloid protein in older adults with Dup15q and autism. And you can see this again through the different, forms of beta-amyloid protein, that there's an accumulation. There really shouldn't be any. And you can see that there is these dark spots in all of these images. If you've heard of beta-amyloid protein, this is one of the many proteins associated with Alzheimer's disease. It's not the only one, and it's not the only cause of Alzheimer's disease. But in many cases of Alzheimer's disease, these beta-amyloid proteins will become really thickened and aggregate and get really sticky and be kind of the cause of Alzheimer's disease. Now, this does not mean that people with Dup15q are more susceptible to Alzheimer's disease. It doesn't even mean that the people in these studies absolutely had Alzheimer's disease, because frankly, Alzheimer's disease really still is categorized by the presence of cognitive deficits. But what it does mean is that people with Dup15q as they age may be more susceptible to cognitive impairments because of this. Now, what do you do about that? Well, the first thing is we know what's going on. I think, again, any sort of intervention that's used for people without Dup15q and Alzheimer's disease certainly needs to be rigorously tested, but at least it's offering some answers about what parents are saying and what siblings, I guess at this point, are saying, is that some of the cognitive functions may be declining as people with Dup15q and autism get much older. So we're not talking about 40s, we're talking about 60s and above. And it can also signal why potentially, and this really hasn't been looked at, but if they start showing cognitive decline earlier. And again, I'm completely in line with the idea that there's not enough studies in adults, but this may provide some specific insights about what's going on. So I wanted to provide kind of like a 500-foot view, or even like a forest through the trees kind of a perspective on all this. First of all, you know, autism related to a Dup15q mutation is somewhat different than idiopathic autism. I'm not going to make judgments about everyone with Dup15q and everybody with autism without a specific genetic mutation, but we do see on the whole some differences in social communication function and some cognitive function. However, we also see outside of that area of chromosome 15, there's not very many functional differences in the genes. The gene activity seems to be on the whole very similar. On the whole, you know, there is the exception to that rule. I mentioned that study about PCBs and chromosome 22. There may be some exceptions to that, but on the whole, if you just take the two together, they seem to be, their genetic activity seems to be very similar, which leads hope for people who want to understand autism without these genetic mutations by understanding Dup15q. And also I mentioned a few targets of intervention. So I mentioned one, the pharmacological interventions for that Fragile X protein, and also maybe for beta-amyloid. What I didn't show you, because I didn't show you because I think I've showed it in previous webinars, is that certain areas of the brain seem to be specifically different in terms of their cell size, and the way that they grow and expand seem to be different. And so that may be a target for intervention for proteins that target those processes of the developing neuron and growing and branching. And then also we're seeing potential neuropathological markers. So what defines the different groups? Can we understand more about Dup15q by understanding what goes on specifically in the brains of people with Dup15q? And the answer is we're getting closer to that. And finally, Dup15q is helping the entire field better understand what goes on in gene-environment interactions. And I think one of the biggest takeaways from Dr. LaSalle's study is that just because there's a strong genetic signal means that there may still be room for environmental factors. And again, I want to define environment very broadly. I don't want people to think just because Dr. LaSalle studied PCBs that she's thinking that it's only PCBs. Sorry, for those of you who don't know about the Autism BrainNet, this is the program that has coordinated all of the collection of the so far almost half a dozen donations from Dup15q families across the country. There's actually now 4 areas, so you will be working, depending on where you live, you would be calling either, there's a 1-800 number that would be called for anything, for any reason, even if you have just a question, and it would be directed to the brain bank that is closest to your location. So I live in New Jersey, so my point of contact would be Mount Sinai, and then people that live, say, in Florida, their point of contact would be the Beth Israel Center in Boston, believe it or not. And then there is a center in Texas for people that live in what I call in the middle of the country, like Vanessa, and then the West Coast. So this has really been helpful in ensuring that not just the donations go smoothly, but also questions are answered, and that there's people in your area that are working with you. And the person that's not on this slide, who's so crucial to this program, is Carolyn Hare. And I didn't include her picture on there, but if you ever have a question, it will probably be answered by either me or Carolyn, so please feel free to reach out to us. What the Autism BrainNet really wants you to understand out of this is not to memorize everything, but the ABN has a newsletter that goes out. We certainly don't expect everyone to say, okay, well, I've absorbed it and I know it. And also, exciting scientific findings are coming out all the time, not just around autism without a Dup15q mutation, but also those with Dup15q. So we have a quarterly newsletter that goes out. If you wanted to sign up for more information, I know some of you have already, which is great. You can go to takesbrains.org/sign up. All you have to do is enter in your name and email address. And I'm sure you've signed, some of you know you've signed up using one of these cards. Literally, and nothing will happen, but you will get this newsletter. I get a lot of questions about, okay, well, if I sign up for the newsletter, am I legally obligated? The answer is absolutely not. The only way a donation is started is if you call that 1-800 number and then you say, I want to make a donation. There's nobody chasing you. This newsletter is only for your own information. You know, science is out there for everyone, not just for the scientists. And so this quarterly newsletter attempts to take the new scientific findings and convey them in a way that is accessible to everyone. These are some of the social media handles if you want to get more information. We're posting constantly on Facebook and Twitter. There's obviously also websites if you have any specific questions about the process. The takesbrains.org site has amazing resources and testimonials from families, as well as where you can actually go and sign up for that quarterly newsletter. So we talked at the very beginning about people who had donated families or people who had donated their brains to research. These are two of the Dup15q superheroes. So their families made the decision after they had passed, and it was obviously both parents who had made the decision to have their children's brains donated for this program. So these are just two of the almost a dozen families, and none of this research is possible without their, commitment to research and commitment to making the lives of the next generation of people with Dup15q better. So I'm happy to answer questions. They absolutely do not have to be about brain tissue research. You can ask me anything you want. If I don't know the answer, I will certainly tell you I don't know the answer or suggest somebody who does know the answer. And sorry, I'll just go back to the beginning on this presentation. Oops. And then, yeah, we can ask questions. I think there's a chat box so that people can ask questions.
Yep, so we're on 2 different platforms right now.
Oh, okay.
I'm gonna go to Facebook. If there's anybody on Facebook who's watching live who has questions for Dr. Halladay, or anybody who's on our webinar, right on the webinar side, feel free to post in any of the questions boxes, and I'm happy to read the questions out loud. I wrote down just a couple things that that kind of came up during that, that I want to point out too, that I think Dr. Halladay went over the sites and the locations and the regions that are covered by the BrainNet, the brain sites. But when it comes down to it, if you are interested or if anything ever happens, that 1-800 number is the best number to call, or even just me. So if you call the 800 number for the Alliance, Carolyn and I, we have a hotline that if there's something going on or if there's a question, we can get, that's kind of the best point of contact. So you don't have to really necessarily worry about those regions, but just know that there's that many dedicated researchers out there who are working on this and are a part of that, and the BrainNet, really. So, and then all the links for to sign up for the ABN are on the website, is our Dup15q Alliance website. So it does link through to It Takes Brains if you want to sign up. So it's for just that newsletter or anything else, or if you have a question. Let's see.
And the number, please feel free to call the number, even if you don't have, if you just have a question, right? So you can just ask a question, and actually the good thing about that 1-800 number is that it will get forwarded not to the actual right person. Like if you have a question about a particular area of research, we'll figure out what scientist is specifically involved with that area of research, and we'll get it answered for you, or we'll loop you in with that scientist if you're okay with us forwarding your email. So it's not just calls for donations, it's calls for anything.
If you see a paper on any of the websites, or if you see a paper that's coming out, like I know that there's been 2 papers recently that have come out through ABN or have Dup15q related to them. But we all know that papers are not necessarily open source publications. So if you want a copy of that, that's another way to get some information.
So absolutely, I'll give you a little trick. So since Autism Science Foundation works with the Autism BrainNet, if you want a copy of a paper, call that number, tell them who you are and that you want a copy of the paper, and I'll try and track it down for you.
Yeah. Cheryl asked, so can you go back to the region map that you showed? They're just
absolutely. Sorry, everybody.
The other thing is, I think the logistics of this are interesting too. That tissue where it lives, it can go back. It's all cataloged, and then where is the tissue actually kept?
So that's a great question. So at each of these sites, there are brain banks. So there's spaces, dedicated freezer spaces, and then spaces in a lab within each of these nodes where the tissue is kept. This allows it so that it doesn't get transported multiple times. And then there's a common protocol. So the brains are treated the same way at UC Davis as they would be treated at Mount Sinai School of Medicine. And we have an informatics system, so each brain has a unique number. Nobody knows your name, nobody knows where you live except for Carolyn, because she does want to reach out to you and check in with you and have contact with you. But when it comes to anybody else, nobody else is, unless you want them to be, knows who you are. And then they're processed the same way. So they're cut the same way, so that when researchers want to study them, they make sure they get the same. If they're requesting this particular region from the UC Davis Bank, they're getting the same particular region from the Mount Sinai Bank. Excuse me, sorry. So in this way, there's 4 main banks, and that's to protect the tissue, and it's also to reduce the the shipping across multiple banks multiple times.
Okay. Adrian has a question. She said, you mentioned about other environmental factors that are concerning other than PCBs. Do you know if they're looking at other factors?
So within autism, that's certainly the case. I hope that let me go back and say, for so long, there has been this kind of belief that because there's this huge genetic influence, that the environment means very little. And in fact, there is a huge genetic component, obviously, in rare genetic disorders, but also in autism, where there's no known established genetic disorder. So we know it's genetic. We just don't know, which particular genes. The misnomer is that it's all genetic or all environmental. And that's where research has really been struggling, I think, and really behind, is that when you see some of these studies that look at genetics and the influence of genetics, of course it's important, but all these analyses have looked at genes alone or environmental factors alone. So scientists are really starting to be better, I think, at understanding how the two interact. And I think Dr. LaSalle's study is really on the cutting edge of this. Now, it hasn't really received as much scientific study as it should. If you think about all the genes out there and all the environmental factors out there, this is like a whack-a-mole, right? You could take this gene and this environmental factor, or then this gene and this environmental factor. So Dup15q and these other rare genetic syndromes associated with autism are really an opportunity to better understand environmental factors. That being said, there are some environmental factors that are rising to the top in terms of their influences by themselves. And these are things like immune activation during pregnancy. So moms who have, what seems to be infection associated with a fever or hospitalization, or, some other, really severe reaction during pregnancy period. Also, there seems to be some consistent evidence around high levels of air pollution. So really, really high levels, where people, live right near freeways, or live in really polluted areas. So, this hopefully will push kind of cleaner air standards forward, at least I hope it will. And then there are also certain drugs when taken during pregnancy, which is shown to increase the probability of having a child with autism. Although a lot of those we're really kind of curbing them back, and those include things like valproic acid and thalidomide. They're not commonly used during pregnancy anymore. But as we learn about what goes on, what's the mechanism, we can maybe predict what other environmental factors, if there's a common biological substrate, what other environmental factors are likely to be risky. But within Dup15q, the only one that's really been studied so far is PCBs. And I wouldn't say that by itself, see, that's the trick, is that by itself, it hasn't been shown to have any sort of interaction with autism. It hasn't really shown in autism to have any gene environment interactions. But when you take that specialized group of people with Dup15q, it does seem to have this interaction. So we need a lot of things within science to make this research go forward. And one of them is better ways to understand the environment.
Thank you. I think that is a great answer. Also, I think that I, in addition to that, I think Dr. LaSalle, who Alycia talks about her research, we are going to actually ask her to do a webinar as well, just kind of because she's environmental factors queen when it comes down to Dup15q and beyond. So we are going to ask her to kind of give an update for her research and do a webinar as well too.
Oh, wonderful. Yeah, let me know, because I would love to sit in on that webinar.
Yeah, because she's just always at the cutting edge and always think she's really wonderful that way, because I think gene environment interaction is so important. So we have a question. Is there a limit on how long the tissue can be stored?
So no, it doesn't seem that way. Clearly we don't want, and the tissue hasn't been sitting around for that long, because for now, the BrainNet gets the tissue and then distributes the tissue. So there hasn't really been any studies within the Autism BrainNet about how long it can last, but we're thinking definitely within 5 years, it can definitely be distributed and used. These fixation protocols really get the chemicals in the tissue, and then it's changed everyone. It's not like it's just put on a shelf and left there. It's really taken care of for the time that it's in the lab, but there's really not a whole lot of. Do you mean the time after it's? So I'm sorry, I'm starting to realize I may be misunderstanding the study. From the time the person has passed away to the time that the brain is removed, or the time the brain is frozen or fixed, put on the shelf, and then distributed. So the answer to the first question is the brain needs to be removed probably within 24 to 36 hours after the person has passed away. That's not a completely hard and fast rule, and sometimes there are exceptions to that. The best thing to do is to call the hotline. And then the Autism BrainNet normally, gets that tissue out to researchers, sooner than 5 years.
She clarified frozen, but I think you answered both of those questions.
Okay.
The other thing is, I do want to mention that for those people who, the Dup15q families who have already donated, like you showed at the end of a couple of the dupers that have already their families have generously contributed, we are constantly, there's always kind of requests for specifically Dup15q brains or tissue, and it's something, it's people even outside of our normal ranks. It's scientists are being attracted to our space because this exists. So we just got a request that is being reviewed right now, and this person is getting support, more financial support to do research on Dup15q syndrome, because this is something that exists, and it's outside of, it's not money coming from the Alliance specifically, but they're getting support because they know that we're partnered this way. So it's something that this is this research can only happen because this net exists, and because it's such great structure that it provides. Also that if you ever, like Dr. Halladay said that, that usually there's a timeline after a passing, but at the end of the day, if you are ever in that situation, you can always call me day or night. Do not worry. It's been, anybody who I've ever talked to that this has happened, you can call me a day or night and we will facilitate this happening in the easiest way possible with the least amount of phone calls for anybody involved, and we make it happen. So please just know that we're here for you, and we can activate this really, really quickly with very, very little that you have to do very much. So just as an FYI. Any other questions? There's somebody from Australia watching too.
Oh my gosh.
Any other questions for Dr. Halladay? I'm looking at both of my things here too. So what we're going to do, so this is, like we said, this is kind of the first, you're luckily our first webinar kickoff in our new series. That's every 2 weeks. What we do is we do record these. It's going to be on Facebook on our parent page, but then we do a webinar recording as well. That's going to go onto our website, and it will also go onto our main Facebook page. So if anybody has any questions, that once we do put this up, the recording, if you have any questions and you post on there, we will bring those questions back to Dr. Halladay and answer them as well as we possibly can that way too. So don't feel like if you missed out on this one and you couldn't get on right away, that all of the questions can be answered later too. And like we'll put up that contact information that Dr. Halladay put at the end of her slides with the post. So
I think at the beginning, sorry, there's the contact information, and then also, yep, if you want to contact me directly with any questions, this is my email address. And if I don't know the answer, I'll find the person who does know the answer. So, or I'll just tell you, scientists don't know that yet. Yeah. So you can call the 1-800 number or contact me directly. The contact me directly is great if you need a scientific paper that you're trying to get a hold of. But you can also call that 1-800 number and the question will get answered.
All right. Well, if there's no other questions from anybody, I don't see any coming in on either one. Just making sure. I think we can conclude the recording. Thank you so much for taking the time.
Oh, thank you. And I'm so happy. And also for those of you I got to meet in Houston, it was so nice meeting you, and I will definitely be at the next meeting. And I know you announced where it was and I already forgot. Where is the next meeting?
We're going to be in Colorado.
Oh, that's right. Okay, great. Yeah. So perfect. So I will see you in Colorado.
Yep, sounds good. And then I have just a question in terms of that follow-up from that conference. I know I took the pictures that I was supposed to send you of everybody who signed up for ABN, but did I send those to you yet?
No, I got the ones. Yeah, it's fine. I mean, you guys who signed up and I got your registration cards, you guys are going to get the next newsletter, which is going to come out at the end of September. But yeah, I mean, Vanessa, if you could send me those, I'll put them into the computer and then everybody will get the newsletter. And it also, if you missed me at the meeting and didn't get swag, which I hope you all did, send Vanessa a note and we'll make sure we get you some swag, either a t-shirt or a keychain or like a plastic cup.
Oh, those by the way, the brain keychains saved my keys. They fell in the lake and it totally fits making my keys float. So thank you for that, because otherwise they would have been at the bottom.
I'm so happy people took them. So I'm so happy. So yeah, I'll put a little package together if somebody wants some good swag.
Awesome. Thank you so much. All right, I'm going to try to close all this down. And we'll be talking to you soon. Thank you so much.
Okay.
And everybody who tuned in. And again, if you're just tuning in and you had questions, please just put them in the comments below and we'll get them back to Dr. Halladay. The webinar too, if you, anybody who registered and who's watching this later, because you're gonna get a link to the actual video. If you have any questions, just respond to that link saying like, oh, I had a question for this, this, and this, and that'll also get to Dr. Halladay. So thank you all for watching and have a great Wednesday and rest of your week.
Yes, you guys too. Thank you guys.
I think the webinar I think I ended it. Did I? Yes, maybe, yes.